Hi guys, this video is for Hinton students in preparation for lab number 2 about photosynthesis. Tomorrow, Wednesday, in class, you can pick up one of these lab protocols from the front table. If you want to pause this video and continue watching it once you have a protocol in your hand, cool. If you'd rather just watch it now, feel free. As long as you've finished watching it by the time you get to Hinton on Thursday, you're in good shape. So I'm just going to go through the protocol to give you a sense of what to expect. So first up, I think that you learned in 9th grade bio the basics of photosynthesis. So you probably learned that water plus carbon dioxide plus light energy is made into glucose, sugar, and oxygen. So those are the basic ins and outs of photosynthesis. You probably also learned that it takes place in a chloroplast, and it takes place in plants. You may not have learned all of the detail that comes below in this picture on your protocol. While we will learn about this in class, you do not need to know all of this information for the lab on Thursday. So a lot of this reading too that goes along with it, you can read through it, but if you're not sure what's happening, it's okay. We haven't learned about a lot of it yet. What you do need to know to be able to follow along with the lab is that all these reactions that happen that we're going to learn about. So we start over here, right here, and we go through here, and the end is over here. So you can see that at the end of all of these chains, there is this NADPH plus right here. And the arrow goes in and it comes out NADPH. So NADP plus is a molecule that can accept an electron. So that's what it does here when it goes from NADP plus. Remember, an electron is negative. It's getting rid of that plus. And also gaining hydrogens and becoming NADPH. So in photosynthesis, this is what happens, but we can't see that with our eyes. We can't see, okay, yes, photosynthesis happened all the way through because there's NADPH. We can't see it. So instead of using, to be able to see it, we're going to use something called DPIP. So DPIP is an indicator, and it's blue when it's oxidized, and it's clear when it's reduced. And what that boils down to is, if photosynthesis has not happened yet, the solution will be blue. If photosynthesis has happened, the solution will be clear. So you'll be able to see that electrons have been freed by what's called an electron transport chain, and they've been sent down to the NADP+, or in our case, to our DPIP. Once the DPIP has those electrons, it turns clear. So again, blue, photosynthesis is not happening. Clear, photosynthesis did happen. So how are we going to do all this? Well, if you flip to the next page, you can see some information about what our goals are and what our setup is. So we're going to be using a spectrometer to measure color change due to photosynthesis. So at the enzymes lab, some of you used Vernier equipment to measure the color or measure the absorbance or transmittance of light through those brown, different shades of brown liquids when you're doing the enzyme experiment. This time you're going to use that Vernier equipment. I think everybody's going to use it to measure how blue, or like on what's how much on a scale of super blue to clear, where your solution is. So like how much photosynthesis has happened. Remember, blue, no photosynthesis yet. Clear, photosynthesis has happened. So you're also going to be studying the effect of light on photosynthesis, studying the effect of boiling plant cells on photosynthesis. You're going to boil some chloroplasts and plant cells and see if they can still do photosynthesis. And then you're going to come up with what you want to test. You're going to do the open inquiry part where you get to figure out what you want to test the effect of. So this has all of the materials and it has a procedure for you. A couple things to note is, again, you'll be checking unboiled. So that's going to be like your plant cells that haven't had anything happen to them, and you'll be testing boiled. Do not add the chloroplasts until it tells you to. So even though on this next page, it tells you what to mix together, make sure you're waiting to put the chloroplasts in until it's time. Because once you put the chloroplasts in, if photosynthesis is going to happen, it's going to happen right away. But you want to be able to track that progress, so you have to wait to add them. You'll be doing all of this mixing in something called a cuvette. These are pictures of cuvettes right here, and I'll just kind of like draw a blow-up image. It's basically a plastic rectangle that you put your solutions in, and this is the implement that the solution needs to be in so that you can test with the spectrometer the absorbance or the transmittance, so like the color, how blue it is or how clear it is. So some other things to keep in mind are there's going to be instructions in your lab protocol that you get at Hinton about how to use the Vernier equipment, how to save the data, how to set it up so that it makes a graph for you, all of that stuff. You also need to bring your lab journal and record your data as it tells you here at step number 13 in your lab journal. So you'll have to set up this table and record the transmittance at each of these times, 0 through 20, for blank, unboiled, boiled, and dark. So for the different conditions that you're testing. So blank is basically what is the color absorbance or what is the transmittance when there's just the cells in there, but there's no DPIP. So what is it when it's like the most clear it's going to be? Unboiled is you're testing chloroplasts that have not been influenced in any way. Boiled, they've been boiled. They've been like cooked. They've been heated up a lot. Think about if you think photosynthesis is still going to happen or not. And you'll also test some plant cells in dark. And again, think about whether or not you think photosynthesis is going to happen. Also, what does that mean? So blue is photosynthesis has not happened. Clear is photosynthesis has happened. So what else to think about? I just want to mention, again, the open inquiry part. So when you get towards the end, once you've done the protocol that it lays out here for you, you get to pick some other condition to test out. So this is a really great thing to ask your tutors for ideas for what to test. Like, what other things do you think could influence photosynthesis? Either influence how fast it happens, if it happens at all, how much it happens, like to what extent. What things could impact a chloroplast's ability to do photosynthesis? So come up with some ideas, brainstorm some things so that you have something ready to test on Thursday. Again, you can ask your tutors for ideas for this. They're super helpful. And I'm excited for the lab. We're going to check out photosynthesis. I'll see you guys later.